Thank you very much, Scott, for that kind introduction. And thank you, everyone, for showing up. And it'll be my pleasure to present to you today. I decided to go ahead and make my presentation going way back, Scott, related to probably even before the NIH existed. No, not that far along, but some personal background. And then I'm gonna go into arterial stiffness, what it is and how we measure it. Then what is the role of the actin cytoskeleton in arterial stiffness? The role of the glycocalyx and how we are trying to join the glycocalyx and the cytoskeleton in how those structures in the vascular wall affect arterial stiffness. And then I'll make some conclusions and indicate some future directions. So I was originally trained as a veterinarian in the National University of Mexico. And at that time, I started working with chickens. And the poultry industry at that time were having a lot of trouble because broiler chickens, let's see, how can I move the pointer here? Broiler chickens, which are the chickens that are raised for meat, were developing what at that time was called ascites syndrome. They were developing congestive heart failure, a lot of liquid in the abdomen, and they were not suitable for commercialization. And this occurred at the end of their lifespan when they were getting ready to go to market. So then it was costing the industry a lot of money. And just to get you, to know exactly what had happened is that from the jungle fowl, they were geneticists through population genetics created a chicken that grew very fast and produced a lot of meat, which was the broiler chicken. And then in this chicken, we started seeing a lot of congestive heart failure. And then the laying hens that were bred from the same background, but they were slow growing, they produced a lot of eggs, they didn't have any congestive heart failure. And we didn't know at that time what was causing this congestive heart failure. So one of the things that I started doing, I participated in trying to see if we could detect this cardiac hypertrophy with electrocardiograms in the chicken. So we were taking electrocardiograms in the chicken and then looking at the mean electrical axis of the heart, trying to determine if we saw any right-sided hypertrophy. We were able to do that. And then we were able to detect which chickens were developing right ventricular hypertrophy from those that were not. And then some people went and used the electrocardiogram to try to select those chickens that grew fast but did not develop congestive heart failure and try to create the next generation of breeders for the broiler chicken and then reduce the incidence of congestive heart failure in chickens. Then I went and did my PhD at Texas A&M University, and at that time I continued to work with chickens. We took some echocardiograms from the chicken, and then we looked at the fractional shortening, and we looked at some regurgitant jets that occurred in the right side of the heart, and we determined that these chickens had severe pulmonary hypertension. They had regurgitant jets that went up to about 60 millimeters of mercury when the normal pulmonary arterial pressure is around 20 millimeters of mercury. And they developed congestive heart failure. They had reduced right ventricular fractional shortening, and they developed severe right ventricular hypertrophy. Then we went ahead and we isolated pulmonary arteries from those chickens, and we put them on a myograph and determined that the pulmonary arteries from these chickens that developed congestive heart failure had an increased hypoxic pulmonary artery vasoconstriction. They were hyperreactive to hypoxia. They had endothelial dysfunction. They had reduced nitric oxide bioavailability as measured by chemiluminescence. And then we also obtained plasma and measured nitrate and nitrite and determined nitric oxide bioavailability. And we also saw that there was some remodeling of the pulmonary artery with hypertrophic remodeling. At that time, we did not measure stiffness, but they were characteristic indices of stiffness in those pulmonary arteries. So now, why is it important to study arterial stiffness and how do we measure it? Well, arterial stiffening, or stiffening of the arteries, is linked to cardiovascular disease. So when an artery is stiffer, when the artery is under passive conditions and we just increase intraluminal pressure, that artery is not able to distend as much as a more pliable artery. And then this decreased pliability or distensibility of the artery increases the afterload in the heart so that the artery is not able to distend, and then the pressure is transmitted downstream. This increases systolic pressure, and also because the artery is stiffer and cannot recoil sufficiently, that also decreases diastolic flow and also causes some problems with coronary perfusion, as I will show you. But more importantly, increased arterial stiffness has been associated with an increased risk for cardiovascular events such as heart attacks and stroke. Now, what is the pathophysiology of this stiffening of the arteries? So in a normal pliable artery, there is a slow pulse wave velocity that is transmitted along the wall of the conduit artery. And this reduces afterload and also allows for a wave reflection. When the wave or the formation of the artery reaches, for example, the iliac bifurcation, there is a return of this wave, a wave reflection, that when it comes back to the heart, it favors the perfusion of coronary arteries and the perfusion of the heart. When the artery is stiff, then this pulse wave velocity increases the afterload in the heart, and then as the reflection is faster, the reflection gets to the heart faster, not allowing for coronary perfusion to occur appropriately. As a consequence, we have an increase in systolic blood pressure, and there is also increased pulsatility that goes especially to those organs that have low resistance, and then it causes end organ damage. So some of the organs that get easily affected by this increased pulsatility are the kidney, the brain, and the womb. So how do we measure this pulse wave velocity? What we usually do in humans is, and also in mice and other animals, is obtain with ultrasound the occurrence of the pulse wave in the carotid artery, and then we obtain it simultaneously in the femoral artery. We measure the distance that is present between the two measurements and the distance, the difference in time on the appearance of the pulse wave. And that way we can calculate the distance that it took for the pulse wave to travel through the aorta into the femoral artery, and then we can calculate the pulse wave velocity. Remember, this is the velocity of the pulse wave of the deformation of the artery, of the wall artery as it travels, not the velocity of the blood. It's probably easier to see it in the mouse when we obtain it with ultrasound. If we obtain B mode ultrasound images and we obtain a line to obtain M mode over time, and we can see how the distension of the aorta takes place. And then if we take it in two different locations and we measure the distance, we can obtain the velocity at which this deformation took place. And then we obtain pulse wave velocity and the stiffness of the aorta. And pulse wave velocity is the gold standard measurement of aortic stiffness in vivo, or arterial stiffness in vivo. Then a seminal article came out in 2013 that showed that even though arterial stiffness had been associated with cardiovascular disease, they showed that in mice that were fed a high-fat diet over time, they did a longitudinal study, and they saw that arterial stiffening or increased arterial stiffness occurred before the presence of systolic hypertension, suggesting that it was stiffening of the arteries that preceded the development of hypertension. And at that time, the prevalent paradigm theory was that stiffening was a consequence of hypertension, that the arteries got stiffer because of hypertension. And So as I mentioned, the paradigm when I started studying vascular remodeling was that stiffening of the arteries was a consequence of increased collagen deposition, extracellular matrix cross-linking, that then caused a reduction in maximum luminal diameter, and that it also was associated with elastin fragmentation. And I included here some of the methods that we commonly use to detect collagen in the artery. We do Mason trichromic stain of fixed and slides aortas. We can also use picrosurious red that stains collagen, and we can use polarized light or not to determine the amount of collagen that is present in the aortic wall or the wall of an artery. And one method that we use continually in our labs is second harmonic image generation with 2-photon microscopy. One of the beauties of this type of visualization of collagen is that requires no staining, and so then it can be determined or visualized in living isolated arteries. So what I'm going to try to convince you now is that over the last 10 years or so, multiple publications have come about that indicate that not only collagen participates in stiffening of the arteries, but also the cytoskeleton, specifically the actin cytoskeleton in smooth muscle cells. And what I'm going to show you data and convince you today is, or try to convince you today, is that stress fibers present in vascular smooth muscle cells, in addition with increased adhesion capacity of these smooth muscle cells to the extracellular matrix, participates in stiffening of the vasculature and in increasing the risk for cardiovascular events. Now that I look down, I can hear that my microphone sounds better when I go this way, but can you hear me well even when I'm okay? People tell me that I'm loud enough most of the time. Okay, so then that increases cardiovascular risk. And I'm going to show you images in which one of the ways in which we measure stiffness is to isolate arteries, pressurize them ex vivo, and then put them under passive conditions, under zero calcium conditions usually, so that they do not actively contract. And then we increase intraluminal pressure, and we see how the diameter increases. And if we look at stiffer artery, it usually does not distend and increases its diameter as much as a more pliable artery. And this is also reflected in the strain-stress relationship with reduced strain. And also, if we calculate the modulus of elasticity, the stiffer artery has increased modulus of elasticity. Please remember that when I started studying this, it was all a little bit difficult to me to understand that we call it modulus of elasticity, and greater modulus of elasticity means that the artery is stiffer, not is more elastic, is stiffer. So one of the things that we did to begin to study the mechanisms and the initial portion of the stiffening and remodeling of blood vessels was to isolate arteries, usually resistance arteries. We cannulate them and we pressurize them and we keep them over time to see whether we could induce remodeling and stiffening of these arteries. So one of the methods that we used to stiffen the arteries was that we cannulated these arteries, pressurized them, and then we exposed them to zero calcium to obtain maximal passive diameter. And then after that, we put them in buffer that contain calcium again. If it was a resistant vessel with myogenic tone, it will develop a spontaneous myogenic tone. And after that, we expose them to vasoconstrictor or other agonists for a certain amount of time. In our experience, we need to expose them to at least 4 hours of vasoconstrictors or other agonists to basically induce some remodeling. After that, we would wash the agonists, the vasoconstrictors, allow them to develop myogenic tone again if they had myogenic tone, and expose them to zero calcium. And when we saw a reduced passive diameter, that usually meant that the vessel remodeled and was stiffer. And if we went ahead and then did pressure diameter curves, we saw again that the vessel had a smaller diameter, it was less distensible, and had an increased modulus of elasticity. So one of the first experiments that we did, Marius Catalin Staiculescu, a postdoc in the lab, did was to induce the remodeling in response to norepinephrine and angiotensin II to try to simulate increased sympathetic outflow and increased RAS activation, and then expose the vessels to a compound that prevents polymerization of actin. Cytoskeleton D is a molecule that caps the barbed end of F-actin stress fibers and does not allow G-actin to come in and increase these stress fibers and polymerize. And what we saw is that when he exposed the vessels that were pressurized and had myogenic tone to cytoskeleton D, these vessels did not change much in diameter. If at all, they tended to reduce their diameter a little bit. But when they were exposed to this cocktail of norepinephrine and angiotensin, they were able to initially constrict, but they were not able to maintain that constriction, and they dilated, and these vessels did not remodel. Remember that I show you that our paradigm is that when we expose them for 4 hours to this cocktail or other vasoconstrictors, we can make them remodel and increase in stiffness. Then Marius did an experiment in which he exposed vessels to this vasoconstrictor cocktail, allowed them to remodel. They had a reduced maximal diameter. And after that, he exposed them to cytoskeleton D, which I told you just prevents actin polymerization. They were already under zero calcium conditions here. And then he also exposed them to this molecule that is macallolide B. And macallolide B, what it does, it actually severs actin filaments. So it actually cuts actin filaments. Remember, cytoskeleton only prevents polymerization, whereas macallolide B depolymerizes the actin cytoskeleton. And then we saw that only when the vessels were exposed to macallolide B, they tended to return to their original diameter, and they reduced the stiffness of the vessel. And we went ahead and did some staining to show that in the presence of cytoskeleton D, there was some patches of actin depolymerization, but in general, the actin cytoskeleton, which is shown here in green, was intact compared to the DMSO control. When he exposed them to macallolide B, it was completely destroyed, so much so that I asked him to stain microtubules to see if we could see other cytoskeletal structures, and then the microtubules were intact. So then we showed that preventing actin polymerization prevents the remodeling. And if they are already remodeled and we destroy the cytoskeleton, then we can de-stiffen the vessels. Then in conjunction with Jerry Meininger and Chongqi Hong, we did a series of experiments in which smooth muscle cells were exposed to a vasoconstrictor or to a vasodilator, in this case angiotensin II or adenosine. And then with atomic force microscopy, we raster the cell and we look at the stress fibers. And then after exposure to a vasoconstrictor, in this case angiotensin II, for 20 minutes, we saw an increase in the amount and caliper of those stress fibers. And when we expose the cells to a vasodilator, in this case adenosine, it reduced the amount and the thickness of those stress fibers. And then Chongqi went about to show also that this occurs very dramatically in the force measurement of the cells if he exposed them to a nitric oxide donor nanoate. And this is important. This is going to be important later. So when he exposed them to buffer, there were no changes in force over time. But when he exposed them to nitric oxide, there was a consistent decrease in smooth muscle cell stiffness over time. So then we started looking, what are the mechanisms then that are causing this actin polymerization and stiffening of the cells? And we looked at the mechanisms that were initiated by activation of GPCRs in response to the vasoconstrictor stimuli that we're putting on the cells. And we knew that they would activate phospholipase C, calcium influx that will phosphorylate myosin light chain kinase, phosphorylate myosin light chain, and then induce contraction. But at the same time, these vasoconstrictors also induce the activation of RAW kinase. And RAW kinase, in addition to inducing a process that is called calcium sensitization by maintaining the phosphorylation of myosin light chain, it also can induce polymerization of actin. So then we say, are we seeing this polymerization, and is this polymerization caused by the RAW kinase pathway, what is causing this increasing stiffness of the cells? So at that time, a little bit before, Eric Backer had published a paper in Circulation Research in which he showed that inhibition of tissue type transglutaminase, or TG2, ameliorated arterial stiffening and arterial remodeling. The argument in the paper was that it was most likely due to a reduction in the cross-linking of extracellular matrices. But then we said, is TG2 activation capable of inducing actin polymerization and the maintenance and fortification of actin fibers, smooth muscle cells, and also participate in the remodeling and stiffening of arteries through that pathway? So just as a brief review, tissue type transglutaminase, or TG2, joins glutamines with lysines, and then that is a cross-linking of the proteins that is usually associated with collagen fortification. But TG2 can also act as a G protein, and then also has GTPase activity and can induce intracellular signaling. So then we hypothesized that there was a problem here with the, it's not a percentage. This is the vascular smooth muscle cell membrane. And then we hypothesized that in obesity and hypertension, there would be an increase in TG2 activity. And we have shown that there is an increased presence of TG2 in blood vessels, and that this would increase RHO-A, Rho kinase, phosphorylate limb kinase, and activate limb kinase. And limb kinase is an enzyme that phosphorylates cophylline. And when cophylline is phosphorylated, is inactivated. When cophylline is not phosphorylated, is active, and it severs actin filaments. So when we thought, if RHO-A and RHO and limb kinase are activated and deactivate cophylline, that facilitates the formation of stress fibers and the stiffening of blood vessels. At that time, it happened that I was working a lot with Dr. Jaume Padilla and Dr. Camila Manrique, and we were working so well together that we decided to join laboratories and form a team that we call Vasorum, and that is focused on understanding the mechanisms that cause arterial disease in diabetes, hypertension, aging, and also trying to see how we can intervene with exercise, changes in diet or lifestyle, or pharmacological therapies to try to address those that increase in arterial disease that occurs in these conditions. So from now on, it is very good and very interesting that many of the results that I'm going to present are basically the combination of our laboratories. So it's undistinguishable. They are not only from my laboratory, but from Vasorum. And so we expanded our hypothesis to include obesity, diabetes, hypertension, and aging. And again, that the fact that we would, with TG2, activation of TG2 would induce RHO activation, RHO, limb kinase, and inactivation of cophylline. So one of the things that we did was try to inhibit this pathway by using a limb kinase inhibitor that is available in the market. And at that time, we were getting omental samples from individuals who were undergoing abdominal surgeries at the hospital. And then from these omental samples, we would isolate small arteries, and then we decided to compare the diameter, the distensibility of arteries from hypertensive and non-hypertensives. And this had been shown before, that the arteries that are obtained from hypertensive individuals are smaller in maximal diameter. They are not as distensible, and they have an increased modulus of elasticity, or moduli of elasticity, indicating that the arteries are stiffer. And we also, Dr. Francisco Ramirez in the lab, developed a system in which he can estimate pulse wave velocity in these isolated arteries, and they also have increased estimated pulse wave velocity. So we went ahead and did some immunohistochemistry of these vessels, and we noticed that they were no difference in the amount of cellularity of the vascular wall, but they have an increased level of P phosphorylated cophylline compared to total cophylline, indicating that cophylline activity was reduced, and that they also had increased amount of F-actin content, but there were no changes in collagen, suggesting again that the stiffening of the vessels was associated with an increase in F-actin content, but not an increase in collagen content. Then we went ahead, and from the vessels of the non-hypertensives, we exposed them to our remodeling protocol, in which we put them for 4 hours in the presence of angiotensin 2 and norepinephrine 2 with and without the limb kinase inhibitor. And when we didn't use, when we used vehicle, when we didn't use the limb kinase inhibitor, the arteries remodeled inwardly. And when we put them in the presence of the limb kinase inhibitors, the arteries actually remodeled outwardly. Remember, these were mostly obese individuals, so this indicates that we were able to make them less stiff, and then that the protocol of exposure to norepinephrine and angiotensin made them stiffer, and then we prevented that with limb kinase. So in order to be more mechanistic, can we do the same thing with our isolated vascular smooth muscle cell protocol? So we expose isolated human vascular smooth muscle cells to norepinephrine and angiotensin 2 in the absence or presence of the limb kinase inhibitor. And then we measure F-actin, and we saw that there was an increase in F-actin after an 18-hour exposure to these vasoconstrictors. And if we incubate them together with the limb kinase inhibitor, we prevented that increase in F-actin content. The same thing happened when we looked at phosphorylated cophylline. Exposure to the vasoconstrictor increased the phosphorylation of cophylline, and limb kinase inhibition prevented that phosphorylation. And when we looked at the stiffness of the cells using atomic force microscopy, then we saw that there was an increase in stiffness of the cells, and then that this was prevented by the presence of the limb kinase inhibitor. So can we do this in vivo? So in order to do that, what Dr. Foote in the lab did was we used these mice that had expressed GFP in smooth muscle cells so that we can observe the vasculature in vivo and then do repeated measures over time. He looked at arteries in the ear of the mice, and we implanted these mice with mini pumps delivering a non-pressor dose of angiotensin, 100 nanograms per kilogram per minute for 14 days, or a pressor dose at 1,000 nanograms per kilogram per minute for 14 days. One of the ears served as vehicle control, was injected with vehicle control, and the other one was injected with the limb kinase inhibitor every 3 days. And what we saw is that comparing day 0 to day 14 of the infusion, we saw that there was a significant decrease in the diameter of the vessels in the control ear, and that this was prevented in the ear treated with the limb kinase inhibitor. And this is the cumulative data. So we see that angiotensin 2 was effective at inducing hypertension at the higher dose, and that this higher dose was associated of angiotensin was associated with an inward remodeling of around 15%, and that this inward remodeling that is commonly associated with stiffening was prevented by the limb kinase inhibitor. So we say, can we intervene in a different region of the pathway? So we decided to intervene in addition to limb kinase to inhibit TG2. And we use a compound, cysteamine, that inhibits the transglutaminase activity of TG2. And we did it in a way in which it would be as a treatment. So what we decided is to use, we have evidence that with 16 weeks of Western diet feeding, we can induce stiffening of the blood vessels in female mice consistently. So then we fed female mice with controlled diet beginning at 7 weeks of age, or with Western diet that contained 46% fat, 17.5% high fructose corn syrup, and 2% salt for 45 weeks. After those 45 weeks of feeding, the control mice remain in the control diet, and the Western diet fed mice received just control water or water containing cysteamine at a dose of 180 milligrams per kilogram per day for 8 weeks. And then after those 8 weeks, we sacrificed the mice. But before, we took measurements of pulse wave velocity with ultrasound. And then after euthanasia, we measure vascular function and vascular mechanics. So one of the things that we observed is that after feeding of the Western diet, we saw a marginal increase in mean arterial pressure that did not reach significance, and that this was somewhat normalized by cysteamine, but none of these changes were significant. But when we looked at pulse wave velocity was increased in the Western diet fed mice, and this was reduced by the treatment with cysteamine, the broad TG2 inhibitor. And then when we isolated aortas from these mice and we exposed them in phase, and then we used the atomic force microscope to measure the stiffness of the endothelial layer, we saw that Western diet mice had an increased endothelial stiffness, and that this was reduced by treatment with cysteamine. We also isolated mesenteric arteries and measured the distensibility of these vessels under passive conditions. Pressure increased, and then when we see the internal diameter, we see that the Western diet caused a reduction in the maximal passive diameter of these vessels, and that this was reversed by cysteamine or increased by cysteamine. And then the vessels were stiffer, as shown here with the elastic modulus, the modulus of elasticity, and that this was in the Western diet, and this was also reversed by the treatment with cysteamine. Now, cysteamine is, we use cysteamine as a non-specific TG2 inhibitor. It has other effects. So then we decided, can we do and show the same with a more specific inhibitor of TG2? And for that, we used a compound made by the company Sedira that is a specific TG2 inhibitor. And we treated mice for 28 days with a mini pump, DV mice. And we showed that the treatment with the inhibitor did not change body weight or glucose tolerance, but it did reduce pulse wave velocity in a set of mice that in which we measure pulse wave velocity. It also reduced the stiffness of the endothelium using atomic force microscopy. And when we isolated mesenteric arteries, it also made the treated animals had vessels with increased distensibility and reduced stiffness. So suggesting that inhibition of TG2 reduces the stiffness in this arterial stiffness in these DV mice. So I've shown you data in which if we inhibit limb kinase or we inhibit TG2 with cysteamine or with a more specific TG2 inhibitor, inhibits the remodeling and stiffening of blood vessels. And I show you data that also indicates that if nitric oxide is used, it reduces the presence of stress fibers, smooth muscle cells, and nitric oxide has been published is a known inhibitor of TG2. So this is where we link it to the cytoskeleton. So what happens? How are we going to link this with the cytoskeleton? So one of the things that we knew and we have shown in many different occasions is that Western diet feeding in mice increases stiffness of the endothelium as measured in endothelial cells isolated or in aortic explants. Then it is also we shown consistently that feeding of a Western diet or DV mice have endothelial dysfunction, as shown by reduced capacity to dilate in response to increasing concentrations of acetylcholine and even on increasing concentrations of insulin as a vasodilator, but they don't show any differences in dilation in response to the nitric oxide donor sodium nitroprusside, indicating that there is, in general, no differences in smooth muscle cell function or responsiveness to nitric oxide. And in a study in which we were looking at the effects of Western diet on the structural characteristics of blood vessels, we looked at elastin. And we saw that there were some changes in elastin, in particular the fenestra, or the holes that are in the internal elastic lamina of blood vessels were changing, indicating that there were some changes in elastin. And at that time with Dr. Christopher Foote, we said, well, is the signaling on the elastin receptor changing because of these changes in elastin? And the elastin receptor is composed of cathepsin A, the insulin binding peptide, and also neuraminidase. And we thought, well, do we see any changes in neuraminidase activity? And as part of the study, Christopher went ahead and measured plasma neuraminidase activity. And we observed that neuraminidase activity was increased in the plasma of mice fed a Western diet. So we said, well, what is neuraminidase doing in plasma? And we started looking at the literature on the effects of neuraminidase. And neuraminidase is an enzyme that cleaves the sialic acids present in structures, primarily structures that are present in the glycocalyx. And the glycocalyx is very prominent in the luminal surface of the endothelium, where it forms these hair-like structures that are known to be mechanosensors for flow, and that in response to this shear stress that flow creates, these mechanosensors transmit and induce a mechanotransduction that produces nitric oxide. So if there is increased neuraminidase activity, we would see a degradation of the glycocalyx and a reduced capacity of that glycocalyx for mechanosensation and mechanotransduction and production of nitric oxide. So that became our working hypothesis, that then we said, well, increased plasma neuraminidase activity should reduce glycocalyx presence in the endothelium. That reduces flow mechanosensation and causes endothelial stiffening and dysfunction, reduction of nitric oxide bioavailability, and as a consequence, increased activity of TG2 and increased stiffening of the smooth muscle cells. So we had previously collected flow-mediated dilation data from individuals that had type 2 diabetes or controls, and we had shown that type 2 diabetics had a reduced flow-mediated dilation, indicating that there is endothelial dysfunction in these type 2 diabetic subjects. So using the plasma from those same individuals, we measured plasma neuraminidase activity, and we observed that they had increased plasma neuraminidase activity, and they had also reduced plasma nitrite as a surrogate of nitric oxide bioavailability. And when we did some correlation of these variables, we see that as plasma neuraminidase activity increased, there was a reduction in flow-mediated dilation. And then as plasma neuraminidase increased, also there was a reduction in nitric oxide bioavailability, and that as plasma nitrite or nitric oxide bioavailability was increased, this was associated with a better response to flow-induced dilation. Then we went ahead and treated isolated arteries with neuraminidase, exogenous neuraminidase that was placed in the lumen of these arteries, and we stained the glycocalyx with WGA fluorescent in green here, and we saw that after exposure to neuraminidase, there was a reduction in glycocalyx content, and that these vessels that were exposed to neuraminidase, in compared to those that were exposed to vehicle control, had reduced capacity to dilate in response to flow induced by the rate of amount of flow that was conducted through the artery. And if we compare it to the amount of shear stress that was generated, we saw this also dramatic difference between the neuraminidase treated arteries compared to controls. In endothelial cells, we looked, well, we are destroying the glycocalyx. Can we see if we are also getting rid of some of the molecules that are usually associated with mechanosensation in the endothelium? So we went ahead and measured hyaluronic acid using a hyaluronic acid binding peptide. And we saw that in response to neuraminidase, there was a reduction in the amount of hyaluronan per cells. These are endothelial cells in culture, which is a known mechanosensor. And then also we saw a reduction in the presence of syndecan-1 in these endothelial cells. Did I say smooth muscle cell before? No, endothelial cells? Okay, the endothelial cells. So in endothelial cells, we saw a reduction in both hyaluronan and syndecan-1, which are known mechanosensors of the endothelium. So we then decided to do a study in which we said, if we treat mice with a neuraminidase inhibitor, diabetic mice, DVDV mice, can we increase flow-induced mechanosensation and flow-mediated dilation? And we decided to use an FDA-approved molecule, which is zanamivir. It's a neuraminidase inhibitor that is usually used to treat influenza. So it's more directed towards the viral neuraminidases, but it also has activity on mammalian neuraminidases. So Christopher developed a system to treat these mice with inhaled neuraminidase inhibitor zanamivir. And this is the way it's used in humans. I don't know if you have ever used it. You inhale the compound twice a day for 5 days to reduce the symptoms of influenza. So the experiment was designed to have the same paradigm. The mice were treated with zanamivir via inhalation for 5 days, twice a day. And then we looked at the outcomes of vascular function. And one of the things that we did first was to measure whether inhaled zanamivir was able to increase the presence of zanamivir in plasma. And we saw that inhaled zanamivir increased the concentrations of the compound in plasma to about 230 nanograms per milliliter. Remember this number because it's going to become important later on in my talk in a couple of minutes. And then what we saw is that when we took plasma from these DVM mice that were treated with zanamivir, we saw a significant reduction in plasma neuraminidase activity, and that we also saw a reduction in aortic endothelial stiffness as measured with atomic force microscopy. And also measured with atomic force microscopy, we saw that there was an increase in the length of the glycocalyx. However, there were no significant changes in pulse wave velocity, so no significant changes in aortic stiffness. Then we isolated mesenteric arteries and we measured endothelial function, and we saw that there was a significant increase in the capacity of these vessels to dilate in response to flow compared to the ones treated with vehicle control, and that they also had an increased capacity to dilate in response to acetylcholine and an increased capacity to dilate in response to insulin, but no changes in the responsiveness to the nitric oxide donor sodium nitroprusside. As in concomitant or in agreement with the results in pulse wave velocity, in these mesenteric arteries, we didn't see any changes in measurements of arterial stiffness. So we said, what if we increase the duration of this treatment with a neuraminidase inhibitor, and also if we can go more specifically with other neuraminidase inhibitors? And for this, we collaborated with Dr. Christopher Cairo in Canada, who has some specific neuraminidase inhibitors. So we placed mini pumps on DVM mice that delivered the inhibitors for 28 days, a neuraminidase 1 inhibitor specific, a neuraminidase 3 specific inhibitor, and zanamivir. And we measured body weight, and there was only a significant change in body weight in the neuraminidase 3 inhibitor with use of the neuraminidase 3 inhibitor, and there were no significant changes in mean arterial pressure. However, when we isolated the mesenteric arteries and we looked at flow-mediated dilation, we saw that the DVDV's controls had a reduced flow-mediated dilation compared to all the other mice that were treated with the different inhibitors, with the NEU1, NEU3, and zanamivir inhibitors, and that there were no significant differences in dilation to sodium nitroprusside. More importantly, after 28 days of treatment, we did see significant reductions in pulse wave velocity with using any of the 3 inhibitors, and we also see reductions in the stiffness of these isolated mesenteric arteries when we use any one of the 3 inhibitors. So can we translate this into humans? So we went ahead and did a clinical trial in which we treated diabetic humans with zanamivir as it is prescribed for the treatment of influenza for 5 days, twice a day. And we observed that after treatment, when we compare the baseline to post-treatment, we saw only an increase of the plasma zanamivir to about a tenfold, one order of magnitude smaller than the one that we observed in mice. So we did see presence of zanamivir in plasma, but it was not as great as that one that we observed in mice. Then when we looked at plasma neuraminidase activity, the treatment with zanamivir was not able to reduce plasma neuraminidase activity. And then when we measure the glycocalyx using the glycocheck, which is a camera that allows for the visualization of the endothelial boundary to that one of the red blood cells, and then therefore calculates the amount of coverage by the glycocalyx, we saw that treatment with the inhibitor zanamivir did not change the length of the glycocalyx, and it didn't either change the capacity of blood vessels to dilate in response to increased flow, which to us suggests that what we need to do is to develop a way in which we can increase the amount of zanamivir present in blood or reduce more effectively neuraminidase activity in plasma. So in conclusion, we have developed this hypothesis that in the presence of some insults such as diabetes, obesity, aging, there is an increased activity of neuraminidase that destroys the glycocalyx. And some of the research that we are performing, we are trying to determine how is neuraminidase increased, and also how neuraminidase is actually destroying other structures from the glycocalyx in addition just to sialic acid. And that this destruction of the glycocalyx reduces the capacity of the vessels to sense flow, reduce the capacity of the vessels to produce nitric oxide. This reduction in nitric oxide bioavailability allows for the activation of TG2, which in turn activates RAW kinase, phosphorylase Lim kinase, deactivates cofilin, and then therefore cofilin cannot sever actin stress fibers, and as a consequence, smooth muscle cells get stiffer and the whole vascular wall gets stiffer. And with that, I would like to conclude and acknowledge, maybe I'm forgetting people to put in this list. I've been very much blessed with many people who collaborate with me, and I'm also very thankful to the NIH, the American Heart Association, the NSF, and of course, the University of Missouri for their support. Thank you very much.
Thanks, Luis. That was great. You've pointed to smooth muscle cells and endothelial cells, both increasing stiffness. In terms of the overall stiffness of the arterial wall, can you assign a greater effect to the smooth muscle or the endothelium? Are they participating equally in making the vessel stiffer?
Right now, we cannot really determine how much the stiffness of one cell or the other affects the stiffness of the wall mechanically. Our hypothesis is that the endothelium being so thin, most likely mechanically does not really participate in increasing the stiffness of the whole vascular wall. But we hypothesize that changing the stiffness of the cell really affects the functionality of the endothelial cell. Therefore, it cannot produce nitric oxide or other vasodilators, or that's the way it affects the stiffness of the whole vessel, not actually mechanically by increasing stiffness, how much it really prevents the vessel from distending. Because of the amount of smooth muscle cells that we have in the vessel, we believe that it's mostly the cytoskeleton in the smooth muscle cells that prevents or participates in the stiffness, mechanical stiffness of the vessel.
And because I know your work, I know you think a lot about the extracellular matrix. And you didn't really talk about the matrix today, but do you think that there's remodeling of the extracellular matrix that's contributing to vascular stiffness as well?
Yes, our hypothesis is that, or maybe it's my hypothesis, I don't want to put it in the mouth of other of my colleagues, but that we initially see these changes in vascular, smooth muscle and endothelial cells functionally, that causes these changes in the cytoskeleton, and this in turn points to the extracellular matrix. I need a stiffer extracellular matrix. So then TG2 at the same time, for example, that it may be stiffening the cell by cross-linking and forming F-actin fibers, it also cross-links extracellular matrices, and then also induces or causes the vascular smooth muscle cell to produce more collagen. So I think that there is avenues of talk between the cells and the extracellular matrix and backwards. So I think that at some point you reach unfortunately, a stage of feed forward in which stiffening of the cells causes a stiffening of the matrix, and stiffening of the matrix also points, tells the cells, I need you to be stiffer, and then that keeps on increasing the stiffness of the cell. And at some point, it is more difficult to de-stiffen a cell that has a lot of collagen and what we would call fibrosis than one that is only stiffened by the cytoskeleton.
I'll ask one more question, if I may. You've selected representative vessels according to the model that you're using, but do you think that these changes that you're describing are happening throughout the entire cardiovascular system, primarily on the arterial side? Are there also changes on the venous side? How widespread is the phenomenon of vascular stiffening?
Yeah, we have looked at different vascular beds in the arterial side. In the venous side, we have not looked at, but the arterial side, it appears to be more or less ubiquitous. We see stiffening on the renal circulation, on the mesenteric circulation, on the cerebral circulation. I did a series of experiments with Sean in which it appears that the coronary circulation may act a little bit different. But in general, most of the vascular beds in the arterial side appear to be similar. Thank you. Yes, Javad.
Luis, very educational seminar, lecture, whatever it is. I have a few questions, but let me ask one funny questions. In TEM images, why Glycocalyx totally black? Why?
Well, we have struggled with imaging with the electron microscope. Why is it completely, shows that with that density? I don't know. We have published some data with EM, but it's really a very finicky technique to try to preserve the glycocalyx. It is difficult, so that's why we have resorted to look at it with the atomic force microscope or with the staining with lectins. But why it is that it shows so dense, I don't know.
I have a reason asking for this question, and important, even though it is funny, but important. How we visualize TEM images for those tissue, whatever it is, that electron beam cannot pass it. We show the shadow of it. Unlike, for example, immunohistochemistry, something everyone knows better than me. So if it is a gel-like structure, thousands of the papers saying Glycocalyx is gel-like structure. How dense is that gel prevented passing electron beam? Or all of these published papers wrong? What is it?
I mean, your guess is as good as mine.
Because I have data to show it is not dark.
It's not dark?
Yes, I don't have it.
Yeah.
Papers, so many papers published. Glycocalyx, one of them you showed, and beautiful images. Don't you think those are, for example, precipitation of the dye?
Some people argue that may be the case, but other people have shown that if you use neuraminidase, for example, to destroy the glycocalyx before and after you obtain, then you see the differences. So if it were precipitation, why would you see a difference with the treatment? So, there are techniques that we need to, but that's why we do it with many different ways. We do it with the glycocheck in vivo, we do it with the WGA staining, and we do it with the atomic force microscope. So I think that when many different techniques show similar results, then we can be more sure of the findings that we have.
Personally, precipitation of lanthanum nitrate.
Thank you.
Sure.
This is just two remarks on this. One is it depends upon the subcellular contribution. I think we had an occasion to look that. So when we scrape the endothelium, the stiffness reduces by about 1 kPa. So the absolute endothelial stiffness is around 1 kPa out of 6 to 7 kPa. So still it has got the importance, it gets altered. And second, the individuality, we had a couple of samples from the vascular smooth KO mice of the Bender's time, we did not pursue further. we isolated a vascular smooth muscle cells from the control and the Western diet. And after the culturing 3 to 4 days, still they maintain their properties. So they are more stiffer, whereas the knockout mice, they are less stiffer. So that individually, there is a difference between the two, but we did not pursue. Coming to the glycocalyx, I also had an occasion. I want to share that these images, I think what he has got images is like a 3-dimensional images after the metamorph or the program, so that when the X stiffness goes in a stake-wise form, as a stake-wise capture, the background of the looks like, because it is a endothelial layer is about 1 to 2 micron is gone, but it is a stake goes on to the deeper, like a 2 thickness, about 25 stakes like that. So it looks like a different way. But if you take the individual cells, it doesn't look like that.
Okay, thanks, Arun.
Hello. I have a quick question about the regional specificity of the stiffness. Do you think is more in the ascending region where you have more shear stress in the arch region compared to the thoracic, or descending aorta, if we take the aorta?
Well, we have used different ways to calculate pulse wave velocity. If that's where one of the ways we can answer that question. Whether we do it with a technique in which we obtain the pulse wave in the carotid and then also in the femoral, and then that encompass the whole travel of the wave through the aorta, we see increased pulse wave velocity. And if we just take a region of the abdominal aorta that you have done it with the vivo, then we saw also increase. So we believe that there is an increased stiffness. Whether it varies from region to region of the aorta, then that remains to be determined. One thing that we do also is we isolate rings of the aorta, and I think right now have been mostly from the thoracic aorta, and we hook them to wires, and when we expand them to determine the stiffness of the vessel, and we also see changes in stiffness. So then we have different ways to estimate stiffness, and it appears to be ubiquitous, occurring at different places, whether in specific regions like suprarenal, infrarenal, it may change and then affect aneurysm development that you study. It remains to be determined. Yes.
Thank you. And one more question. Have you seen any changes in the proteases that target the cytoskeletons, like calpain or cathepsins, which are the intracellular protease? Because you're talking only on the cytoskeletal disruptions. I'm just curious about that.
Well, we have looked at calpain, and I don't remember right now any results, but we have looked at ADAM17. And ADAM17 activity is increased in diabetics, and we have shown that it participates in inducing endothelial dysfunction. We believe it sheds multiple components of the surface proteins on the endothelium, and most likely in smooth muscle cells as well. So it is involved in AAA. So
yeah, we have the calpain mice in our lab. I will come and talk to you. Thank you.
Thank you.
Well, thanks for your attendance today. Let's thank Luis for a great talk.
Thank you.